Hello, this is your lecture on tissues. We have completed the chapter on cells, so now with the next level of organization, cells come together that are similar in structure and function, and they make up tissues. So we'll get right into it. The study of tissues is called histology, Histology, and in the body we have 4 tissue types and 4 tissue types only. So what I like to do is to present a bit of an outline in regard to how we are going to proceed identifying those 4 different tissues and what it is we need to know in regard to our embalming process. So I've outlined them here. The tissues that we will be identifying include epithelial tissue, followed by connective tissue. And under connective tissue, we have subcategories. The subcategories include the connective tissue proper, supportive connective tissue, and vascular connective tissue. Under the connective tissue proper, in your textbook, it goes over several different types of tissues within the connective tissue proper. We are only going to focus on adipose tissue, as you see outlined here. When we move on to the next type of tissue under connective tissue, we will focus on supportive connective tissue, and the 2 subcategories include cartilage as well as osseous tissue. Osseous tissue is what we commonly refer to as bone. And then finally, we will identify vascular connective tissue. And under vascular connective tissue, even though I haven't listed it here, vascular connective tissue would be blood. And we will also go over the components of blood. And the final 2 tissues that are included are muscular tissue, of which there are 3 types. We have skeletal tissue, we have smooth muscle tissue, and we also have cardiac muscle tissue. And we'll identify and compare and contrast the 3. And then finally, we have nervous tissue. If you look in Blackboard, if you have not done so already, there are some handouts that you will need to print so that you will understand what I'm reviewing as I go through this video lecture. On Blackboard, there are blank copies, and there are also the copies that I'm actually going to be utilizing in this lecture. So you'll have them both, one that has writing and then one that does not. The ones that do not, that's for you to practice and to help edify your understanding of the material that's being presented. Okay, so as we go forth, we're going to do them in the order that they're listed, that I just had them on my whiteboard. And we'll start with epithelial tissue. I'm going to erase the whiteboard just in case I need to write on it later. And if you could, and if you're going along in your textbook as well, you could just identify the sections in your text that you will need to read and make sure that you have an understanding. If I don't cover it in this video lecture, then that means you're not responsible for knowing that information. But starting with the first diagram that you have, it looks like this. This is obviously the one that has the writing on it, but there's also a clear document as well. Okay, so we're going to start talking about epithelial tissue. Epithelial tissue, obviously, as any tissues are, they're made up of cells. And we're going to identify the different types of cells that compose epithelial tissue and determine how we actually classify and we name those tissues. All right, so on your document, you should be able to see that it has bracketed off the tissues as such. We have what's referred to as squamous or squamous. Next, we have cuboidal, followed by columnar. Now, these are identifying the different shapes of the tissues. And you'll notice that at the top of the diagram here, it identifies not only the shapes of them, but it also shows you the layers. We'll talk about that in a second. To keep this up here, just a moment. Okay. You will also notice that there's an area that I have actually, in colored pencil, made it like a magenta type of color. That's called the basement membrane. The basement membrane is where all of the cells are actually attached. And from looking at the basement membrane, we can also determine the layers of the cells. So starting with the cells up top here, we have the squamous cells. Squamous cells are flat cells, F-L-A-T. So under that word, just put flat so that you will know their shape. And here is indicating we only have one layer of flat cells that are attached to the basement membrane. Therefore, the name of this tissue would be called simple, because it's only one layer, squamous epithelial tissue, or squamous. However, in the next diagram, you will notice that here's the basement membrane, but there's more than one layer that's attached. It can be 2 layers, it can be 22 layers. As long as there's more than one layer, the name of the tissue is then referred to as stratified. So if I find my basement membrane, I see more than one layer of flat cells attached to, or well, more than one layer of flat cells on top of the basement membrane, then the name of the tissue would be stratified squamous epithelial tissue. So basically, if I had to kind of create my own bit of an equation, so to speak, to help me identify tissue, I would do it like this. If I were to look under a microscope, I would create my own equation, which would be the layering plus the shape equals the type of epithelial tissue. Number of layers, shape of the tissue, and then add epithelial tissue at the end. So then for the next box, the shape of the tissue are cubes. It's cube-like, thus the name is cuboidal. So here, attached to the basement membrane, I only see one layer. So based upon the equation that I just created to help me identify the different types of tissues, I would refer to this as simple. They're cube-like cells, cuboidal, and then epithelial tissue. For the next grouping of cells, I see more than one layer that's atop the basement membrane. So more than one layer would be stratified. The layers, stratified, the shape, cuboidal, epithelial tissue. Simple enough. And then finally, I have column-like cells. So these column-like cells are referred to as columnar. Once again, identifying the location of the basement membrane, I look to determine how many layers I see. I see one layer attached to the basement membrane. Therefore, the name would be simple columnar epithelial tissue. And then without even looking, we can figure out what it would be called if there was more than one layer of column-like cells. That name would be stratified columnar epithelial tissue. Now, if you'll notice on the document, I've kind of created another basement membrane over here because the basement membrane actually has a superior layer to it and an inferior layer. The superior layer of the basement membrane is where we are looking for the point of attachment of the cells. So that superior layer is referred to as the basal layer, while the inferior or the under layer is referred to as the reticular fiber layer, as listed on the document. Now you'll notice that there's one last grouping of cells right here. But you'll see that this right here is actually indicating that it's the basement membrane. This is the basement membrane. But we start with one type of cell shape, and then it changes to a different type of cell shape, and still another one. Thus, the name of this tissue is called transitional epithelial tissue. Now, transitional epithelial tissue, we don't have to indicate a layer because the name transition itself is telling you that it changes. So if it's changing from one thing to another, thus more than one layer. The name transitional epithelial tissue. In your textbook, it also identifies one last layer or epithelial tissue that you need to know, and that's called pseudo-stratified epithelial tissue. The prefix pseudo means false. Stratified, we already know, means more than one layer. So the word pseudo-stratified is telling you it's falsely looking like it's more than one layer. If you look in your textbook and you look at the diagram that they have in the text that identifies pseudo-stratified epithelial tissue, if you look very carefully, you will notice that actually each one of those cells does touch the basement membrane. But because of the positioning of the nucleus that's inside of that cell, inside of the cell, it almost looks like it's alternating to give the appearance that it's layered. But upon a closer look, you can clearly discern that each one of those cells is reaching the basement membrane, thus the name pseudostratified. Okay. The next type of epithelial tissue that you'll identify will be under the connective tissue proper, and that's adipose tissue. Adipose tissue is made, and adipose means fat, so adipose tissue is made up of fat cells. A fat cell is called an adipocyte. We talk about adipocytes because adipocytes and adipose tissue has a tendency, or it has the ability to create what we refer to as a secondary dilution of our arterial fluid. So the word secondary itself is indicating there must have been a dilution before that, because secondary is the second one to occur. The primary dilution is as such, and I'm going to give you an example before I tell you exactly how it happens as far as the embalming process. If you decided to go to the grocery store and make some orange juice from the concentrate, you would go to the freezer section, select the type that you wanted, and it comes in a can about like this. But you don't drink it like that because it's concentrated. What you have to do is add water to it in order to give it, for it to have its desired taste. That's the first time that you add water to it. That's a primary dilution, because I took the concentrated formula, added water to it. Only one time though. So now you've gotten your orange juice to taste just like you want it, but you want it a little colder. So then you add ice to it. When you add the ice to the already diluted orange juice, but it's to taste currently, if you allow the ice to sit in the fluid and allow it to melt, it's going to alter or augment the change, augment the taste that you originally developed, because your primary dilution, when you first added it to the concentrated orange juice, was perfect. But then when you added the ice and the ice began to melt, it made it more dilute. So the ice created a secondary dilution of your orange juice. Now, I gave you that example so you can further understand it in regard to embalming practices. Cases that we have that have larger amounts of adipose tissue have the potential to have a lot more water in their tissues because adipocytes are very large cells. And they're so large and they are so full of water that the nucleus most of the time is pressed up against the cell membrane because the rest of the cell is filled with water. Now you might be thinking to yourself, well, then that would be a primary dilution of our fluid. No, it would not, because we actually have to go through the process of mixing our arterial solution. What we do is similar to creating some orange juice that you get from concentrate. We take our arterial fluid that comes in a bottle, 32-ounce bottle, and it's concentrated. We take that concentrated fluid and we put it into our embalming machine that may have 3 gallons of water in it. So that's my primary dilution right there. When I have the water in the embalming machine and I'm adding my concentrated arterial fluid to it. Well, I turn my embalming machine on, it will mix it up. But then at that point, I have to take that same fluid that I've gotten just right and inject it into my case. Well, if my case already has a lot of fluid in the tissues, it could potentially dilute the fluid that I've already created, just as the ice would dilute the orange juice. So we always have to take caution and just be aware, because it doesn't happen all the time. But there is the potential that exists that and more of an amount of adipose tissue, the more adipose tissue that a case has, it could potentially create a secondary dilution of our fluid. Hopefully that helped you understand it a little bit more. Okay, so adipose tissue is the only one that you need to know under the connective tissue proper. Once we move beyond the connective tissue proper, then we have 2 other types of tissue. The first one is cartilage. Now, you don't have a diagram for cartilage, but I am going to put up here the 3 types of cartilage that we have. We have a very flexible type of cartilage that it's like the tip of our nose. The tip of our nose is actually made of this type of cartilage, and it's called hyaline cartilage. There are actually a few parts in our ear that might have a bit of hyaline cartilage as well, but hyaline cartilage is the most flexible of the 3 types of cartilage. And like I said, you can find it right here, tip of your nose. The one that's moderately flexible is elastic cartilage. Now, we can definitely find elastic cartilage in our ears. Some of the portions of our ears that are not as flexible as the ball of our nose, the protruding lobe of our nose, that's actually elastic cartilage, such as the antihelix, which is right inside of the inner rim of the ear. And the least flexible cartilage of them all is called fibrocartilage. Fibrocartilage is designed to not move very much. It may have a bit of it may be able to move a bit slightly, but it's not designed to be as flexible as hyaline or elastic cartilage. We can find an example of fibrocartilage between our 2 pelvic bones. Our pelvic bones are called the os coxa, and they actually meet anteriorly in a region that's called the pubis. In between those 2 bones, we have what's referred to as the pubic symphysis. The pubic symphysis is made of fibrocartilage. If you will take out the diagram that looks like this, it's referencing osseous tissue. Osseous tissue is also commonly referred to as what we would say bone. If you're going to see an osteologist, they study bones. And thus we have actually a picture of what's referred to as an osteon. you can kind of see concentric rings. They almost look like if you had cut a tree down, you'd see the concentric rings inside of a tree. And there's several of them that you can actually see here. This is a slide of compact bone. Compact bone is what actually makes our bones rigid and hard, but we do have another type of bone that's referred to as spongy bone. Spongy bone is also referred to as cancellous bone, C-A-N-C-E-L-L-O-U-S, cancellous. And cancellous bone can be actually found at the ends of our long bones. It's where you'll find that's also areas of red bone marrow where red blood cells are made. And we'll talk a little bit more about that when we actually get to the skeletal system. But this is actually a slide of compact bone. At the bottom of this, I've kind of shown you where you can find the different types of bone. The ends of long bones, this is my best diagrammatic representation of a femur, which is your thigh bone. Might not be very good. Like I said, it's my best diagrammatic representation. And I've identified the ends of the long bones as the epiphysis. That's where you'll actually find the red blood cells and where hematopoiesis occurs, which is the formation of red blood cells. Hopefully you can see that. The shaft of the bone is referred to as the diaphysis. Now, the diaphysis is actually where we will find compact bones, such as we see here. A bone cell is called an osteocyte, and they are produced inside of the shaft or the diaphysis of the bone. The other information that you have up here as far as identifying different parts of the osseous tissue, you're not going to be required to know. But what I do want you to understand is that each one of these little black areas here, not the dark and blackened circles in the middle, but these little dark areas right here, each one of those is actually an osteocyte inside of the concentric ring, which is the osteon. So there we have our supportive connective tissue. And then finally, under connective tissue, the diagram that you have that looks like this. At the top, we have vascular connective tissue. Vascular connective tissue is blood. Within our blood, if you actually centrifuge it, which means if you took some blood, put it into a test tube, put it into what's referred to as a centrifuge, it spins the test tube very, very fast. And the formed elements, which are the cells that are inside of blood, will settle at the bottom, and there'll be a very dark red, almost brownish color. And to the top will rise what is referred to as plasma. Plasma is a straw-colored fluid. It kind of has, I just took something out off my table, kind of this color to it. That's straw-colored. And it's composed mainly of water. And plasma is designed to nourish the formed elements. So inside of our blood, we have red blood cells, white blood cells, and platelets. Red blood cells scientifically are called erythrocytes. White blood cells are called leukocytes. And platelets are thrombocytes. They all have specific functions. Red blood cells are designed to transport oxygen. White blood cells are designed to fight infection. And platelets or thrombocytes are designed to promote clotting or blood coagulation. In this diagram that you have, it's actually a blood smear. What you see most numerous in number are the red blood cells. Red blood cells are biconcave disc and they have no nucleus. We have two different categories of white blood cells. White blood cells can either be granulocytes or agranulocytes. The granulocytes typically have a bi or trilobe nucleus, or one that looks like it's segmented, like you see here, like you actually see here. And if you look very closely at them, they look to have granules inside of the cytoplasm, which is in contrast to the agranulocytes. The agranulocytes typically have a very, very large nucleus that almost encompasses the entire cytoplasm inside of the cell. And there are two distinct types, and they are typically the larger of the white blood cells. Monocytes and lymphocytes are agranulocytes. while the granulocytes are basophil, neutrophil, and eosinophil. And then finally, there are little dots that you can see throughout the blood smear itself. If you look very carefully, like here's one here, there are a couple here, and I think my arrow is actually on top of the one that's right there. Those are the thrombocytes. The thrombocytes are just cell fragments, and what they will do is, in cases when we begin to bleed, the thrombocytes will get to that area and they literally stick upon each other in order to clot so that we don't hemorrhage to death. So that's our vascular connective tissue. Moving on to the next tissue type of the four, we have muscle tissue. We have 3 diagrams at the bottom that identify the different types of muscle tissue that we have. Muscle tissue is either categorized as skeletal, cardiac, or smooth. Under each diagram, I have shown you the differences, or beside each diagram, I've shown you the differences between the 3. I'll explain some of what's listed here so that you'll understand what you're looking at. If you look at skeletal muscle tissue, and if you look at this diagram very closely, you will see what look like little fine lines running through the tissue. Those are striations. And also, these tissues are running parallel to one another. So, for those reasons, we say the skeletal muscle tissue is striated. In addition to that, it's not branched because the tissue runs parallel to each other. It's multinucleated. If you look a little bit more closely, you will notice these darker areas that you see in between here and here, maybe one up here. These are nuclei, and there are multiple nuclei throughout the tissue. So therefore, we refer to it as being multinucleated. And finally, it's voluntary. Voluntary means we have the ability to move it. Skeletal muscle tissue is attached to our skeleton. So our skeletal system works in conjunction with our muscular system to allow for movement. Anytime we have the ability to control it, it's referred to as voluntary. If we do not, it's referred to as involuntary. The other 2 tissue types that you see are identified here. This is identifying smooth muscle tissue, and you can see the various characteristics of smooth. And then finally at the bottom here, we have cardiac muscle tissue. Now, what differentiates skeletal muscle tissue from cardiac muscle tissue, it's not the striations because both of them are striated. Obviously, with cardiac muscle tissue is found in our heart. So if we think about that, we do not have voluntary control of our heart. So cardiac muscle tissue is involuntary. And in addition to that, just by looking at this picture, you can see how it branches and it's not parallel as we see with the skeletal muscle tissue. One other identifying factor that you can see with the cardiac muscle tissue is that it has what's referred to as intercalated discs. The intercalated discs actually separate each cell within the tissue. So if you look very carefully, you can see how these 2 intercalated discs separate this cell from this one. And you can actually see one nucleus per cell in the tissue. And finally, the 4th tissue type that we have is nervous tissue. A nerve cell is called a neuron. And we have a picture of a neuron right here. It doesn't look like any of the other cells that we've identified. It almost kind of looks like an exploding star. And the neuron actually is responsible for transmitting impulses to allow the body to function. Our nervous system controls everything it is that we do. So here are the parts of the neuron that you need to know. Here, the cell body where you will find the nucleus is also referred to as the soma. Attached to the soma, we have what are referred to as axons and dendrites. The dendrites are the smaller projections that are off of the soma, and they are responsible for bringing nerve impulses toward the soma. While this long, thin tail that you see here, and if you actually look a little bit closer, it made it a little larger. In that very, very long thin tail, you'll see that fiber that's running down. That's the axon. Axon starts with A. It takes nerve impulses away from the cell body or the soma. Also, I have put a bracket around what's referred to as the axon hillock. The axon hillock is where the axon actually meets with the soma. There's no delineation, there's no line, or there's no breaking point. But that region right here, where the axon meets the soma, right there, is the axon hillock. In addition to that, you notice I have kind of put in some dots around the neuron that are kind of floating around in what's referred to as cerebrospinal fluid. These are neuroglial cells, and the neuroglial cells are designed to nourish the central nervous system. Our central nervous system consists of our brain and our spinal cord, and they are actually what keep everything about us going. So they have to be nourished. Okay. Well, if you have any questions, feel free to contact me. This concludes your video lecture for tissues. Have a wonderful day.